here, sort of it gives an example of the kind of collections we have here. It's we have a limited amount of floor space, so we've built up. We have these various tall collections. So we have these long staircases on wheels that we can wheel around to access the top collections. What you see out here is actually sort of miscellaneous material. It's not really part of the main collection. It's things that are collected but unsorted, or sort of bulk collections of things like, a big net came through and dragged a bunch of little plankton and that's preserved, put away, or some things that are part of projects that are being worked on, or some things as part of like a backlog that we don't have proper data for. The rest of the material, the real material, is actually in the collection, which is just through here. So we'll just walk through here briefly.
You could leave your stools and then and you'll come back to them.
Okay.
Yeah, right? It could be.
So feel free to come on in.
Oh, I feel like I'm moving.
And so this is the main collection. Again, to save space, we have these big collapsible shelves, which are really handy. It's sort of like a big libraries or big archives. You see similar things. And like a library as well, you have to have a good system of organization. We have about 65,000
So I'm just going to scoot through
here, in our invertebrate collections. So
if it's possible, just to scoot forward just a little bit, just so because.
So like I said, we have about 65,000 jars, which is a lot to take care of and a lot to keep track of. So of course, there's far more than you can possibly keep in your head at any one time. So it's organized and cataloged in database like you would for a big library. I mean, it's sorted in a way that it's done taxonomically, almost like you would sort by topic in a library. Like we start with, sponges, and then go to jellies, and then worms, and then crustaceans. And so we're here in part of the crustaceans, the Anomura, things like hermit crabs, and then we switch over to chitons and mollusks behind you, and that'll continue on through. And controlling all of this, or on top of all this, we have a big computer database in a special software. So if someone is interested in a particular specimen or a particular species, I can go to that software and I can type in it's a certain number or a species and say, okay, show me all of this species, or show me all of this species from this area, or hey, I found this specimen and it doesn't have any data but it has a number. Show me everything else that was collected in the same trawl as it. Maybe they have data on them.
Do you do this longitudinally as well over time?
Yes, yeah, we do. So we can collect all that kind of information. So this is a good example of this is a nice big crab. It's called Philolithodes papillosus, and on this label, gives us all the kind of information we want. We have sort of a little taxon code, that is used sort of to sort of categorize it, almost like a Dewey Decimal thing. Like 25 is crustaceans, 6 is Anomura, 11 is the fat, so on. Then there's a unique collection number, 973171. We see how many specimens are in the jar. It has a species name. We know where it was collected, Arbutus Island. It was collected by, I think, Bruce Cook and Alex Payden. Alex Payden was the old fish curator, who identified it and when. And then there's even more information, visible, on the database. I can go in and type in that number, or search for this crab and show up, all the other ones. And that happens, from time to time, quite a bit. Like I had someone who wrote to me from New Zealand and said, I'm doing a study on this particular kind of squid. Can you tell me how many of them you have in your collections? So I could search for them and give him his big list. He said, that's great. I'd like to have this on loan, please. And like a library too, we will take, we'll take the specimens out of the jars and we'll wrap them up very carefully and put them in bags so they won't dry out, and we send them through the post. And that's how museums work, and they have to be, because if you're a scientist doing a big study, you either have to travel around to sort of all the world's big museums and see all their collections, or you get material on loan, or you do both. In this case, I mean, I think he had seen some of this material as well, and then now since he's seen it, he's made notes on everything, and then says, I'd like to have this back, on loan to him. So we'll send it off to New Zealand for a loan, and he'll use it for his study, and he'll send And that's
is it true, Hugh, that they also send back the information that they
They share.
they
Yes.
worked
that's
on,
true.
so that
Any
it
kind of
adds to our collection?
If they've re-identified it or updated it, then yes, that all happens as well. And that's
Hugh, may I
important.
ask something?
Yes, of course.
We had a cart for the tide pool, and our gumboot chitin, of course, went walkies
Mhm.
or ended up in the pool by a child, we're assuming. We don't have anything that we can talk to people and actually show them the underlying carriage and how it is and all the things like. Do you have anything in that?
Any spare gumboot chitons?
Yeah.
I can't say. I don't know if I do.
I know I hit a nerve, but really and truly talking about them, and we've got other things. Not a lot on the cart, really. It really needs to be updated.
Oh no, of course, yeah.
But just to have it, we could just pick it up and say, this is how it is, and then, you know.
Was it a model or a dry one? Or
It was a dry one.
Okay.
Yeah.
Well, it's a rehydrated one now.
Yeah.
We have a sea star, right?
Yeah, yeah.
That we can turn
We can
upside
turn
down.
upside.
Yeah.
But the kite was something that most people have never heard of or
Yeah.
seen. And I'd like to get one back, please.
Yeah. Okay. Well, we can try. We could always try and collect one and dry it out.
Yeah, that's
So
what we
I'll
want.
mention it to him.
Yeah,
Yeah.
please.
We have to go figure out where we have some. Yeah.
Yeah.
How often do you change the ethanol?
It's an ongoing job. Generally, we don't have to do it very often. Every 5, 10 years. I mean, it's usually we go through and we top up. Like we go through and we test. Is it still at its concentration? You know, it's about 70% ethanol to preserve things sort of indefinitely.
The reason I'm asking is because Gavin, he was telling us that he touched one of the jars up in the marine room and it split.
The jar.
Yeah, the
Wow.
whole thing opened, so I don't know
Was
how
it a
long
plastic jar?
I have no idea.
No, it was glass.
I don't
No,
know
they're
how
glass.
long,
Yeah.
how long they've been up there.
Yeah, that's impressive. I have no idea. No, these ones are sort of modern glass, and generally the idea is, you know, we don't have to look at things that often, because even just something as simple as topping up ethanol when you've got 60,000 jars becomes
Oh my
a
God.
huge becomes a huge job. So the idea is you should be able to sort of put it away and have faith that it will be fine for many
You
years.
don't keep track of it on the computer.
Sometimes we
It
do.
lights up and says it's time to change
No.
something.
No, we just have often like a volunteer who starts at one end of the collection, and then, you know, just sort of goes through and tops things up, and they make a note of when it was last done, you know, sort of on the edge of the shelf, and then just as they go through, they can, you know, when they get to one end, they start, by that time, it's probably years later, they can go back to the beginning again. So generally, you know, just so long as the level is good. Yeah. Yeah. Usually, if the level of alcohol is good, then that's good. Like that's a good indication that none has evaporated, so the concentration is the same.
We just want to make sure that the specimen isn't exposed outside of the liquid.
Yeah, and we don't want them to dry out, especially soft specimens.
Do you have any idea how old your oldest specimen is?
We have several things from the 1800s. Yeah, and so, I mean, the idea is that these things last indefinitely. I mean, I've seen specimens from the 20s, from the 1920s. I've seen specimens from the 1800s. I've seen specimens that were collected by Charles Darwin. You know, so it's
Not in this collection.
Not in this collection.
Oh, no.
In other machines.
When you were in Australia, did you have anything to do with shark bait and the strombolites?
No, unfortunately, no. I was on the wrong side of the coast.
Okay.
So I was doing some interesting deep sea work on the south coast, which was interesting, which was a lot of fun. We did a lot of interesting collections, going out to sea. Yeah. So the day-to-day of what we do down here is, well, it certainly keeps you busy. There's lots of varied tasks. It can be things like handling loan requests, requests for data. It's not usually just going through and topping up things, but even as simple as I have a new specimen of something that it's gone through committee, I have to register it, put it through the database, make new labels, find a place for it, and oh, well, the shelf is, you know, maybe this thing is full. Well, then maybe what I have to do is take a look at some of these old ones and say, can they go with the smaller jars? And so even something as simple as just putting one jar back on a shelf, you see a lot of these ones are actually pretty full. So that sets off a cascade of other things that need to be then done to help, you know, make sure everything goes back on the shelves again. Or you might, you know, do clean up this jar or this one here, or you have to move a spare one and move a jar over to the next one over, and then that kicks off down the line. You know, so even fairly simple things like that, yeah, will have an effect.
So right now, Henry Chung, the curator, is out on the C3 ship, on the ship that's going around Canada. And we were talking with him yesterday on Google Hangout.
Oh, great.
Yeah, and he was showing a sea star that he just collected that I'm assuming will come into the collection here.
It'll be exciting.
So now there's this big sea star, and then you have to find a place to put it, right?
Yeah. So it goes.
Room here.
Yeah, right?
So it goes through several processes where, I mean, we only take, nowadays we only really take specimens if we know, like, if they're good enough. We do want to collect, the same species again and again. That's good, but we have certain standards in terms of, like, we want good data associated with it. We want to know exactly where it came from, who collected it, when. We don't want somebody that says, oh, here's a jar, here's a specimen. Because if we don't know where it's from, what depth, when, it's actually fairly useless in terms of scientific research and in terms of, like, influencing biodiversity studies.
Do you take
Unless it's a
live
gumbo
specimens?
chitin, then we'll take it.
And then
Sometimes if we out in the field, we collect specimens and they're live, and then we put them to sleep and preserve them.
Oh.
Yeah, we don't usually find stuff when they're like find it dead and then collect it. We usually collect it fresh and then.
That was another question.
Yeah. And some of it is stuff that's collected, people sent in or donated, but mostly we get stuff that's from other researchers or because they're finding stuff that's more interesting. And just before we go back into the lab, I will comment on what we have in this collection is, it's not just stuff to be put out on display. It's not just stuff to sort of, make tick off a list and go, okay, yeah, we've got one of those. It's an archive, and it works because we have collections of many different species from across the province and across time. It's not Noah's Ark, where we go, okay, or a stamp collecting exercise, we go, okay, well, we've got one of those species, we don't need any more. The whole point is you go back and you collect again and again and again, because species change over time. Their size changes. They have different variations. Maybe what you thought was one species turns out to be different species in the north part of its range or the south part of its range. Or, there's lots of interesting kind of scientific questions you can go back and answer. You don't know necessarily at the time, but if you have access to a data set like we do, you can answer all sorts of interesting questions. Like, well, hey, we have this species or this area, and an invasive species came in. Well, we have specimens before that invasive arrived, and we have specimens after. How did that affect their body size or their shape when that competition? Or maybe now we've got analytical equipment that can detect heavy metals in preserved specimens. Well, we've got a record back 100 years in Ladysmith Harbor, so we can go back and we can check. We can actually, so you can do really interesting stuff like that, and it only works because we collect stuff again and again and again. And it's just to show it's not a just a one-off of everything. Museum collections have to grow. They have to continue, otherwise they're useless. It's like if we stopped doing the Canada Census, they tried to, and it was a terrible idea because it only works if you have good data again and again and again. Otherwise it's just a time capsule.
Do you ever have to destroy anything in here? Any specimens because of decomposure or something like that? It's just not.
Occasionally, and we have to go through a whole process.
Yeah.
It's called deaccessioning.
And how would you dispose of them? How would you get rid of them?
Often because they've already been treated or preserved in formaldehyde or alcohol, they then go away as hazardous waste. They go for like a waste disposal. Nothing goes sort of into the garbage. In fact, even if we have something like a fresh specimen that doesn't get preserved and doesn't get put into the collection, like Henry brought in some oysters that he collected some and wanted to look at them, and I won't put those back in the water because once it's been in the lab, it can't go back in the wild again. I won't do it, and I won't put them in the garbage either. So they have to be treated as hazardous just, I mean, it's just a covering thing. I mean, what if, you know, we have some, what if they picked up a virus from old specimens that was lying dormant? Who knows? You know, it's a matter of biosecurity and care.
Good for you.
And thank you. And for all specimens, if they're particularly damaged or they have no data, or in one case we found specimens here that actually belonged to another museum, and they were erroneously registered here. Well, then we have a process to sort of to get them de-accessioned.
And occasionally for the different departments, if there's no data, then they'll give it to the learning department for a handling collection.
So most of our handling collection comes from specimens that have no data associated with them.
But if a specimen is lying when it's collected, it's never put into our ocean station though. The
I don't
animals
know.
that are in
I
the
don't know.
tide pool in the ocean station,
I
they
don't
just
know.
come strictly from the lab? Is that?
No, not from the
That's
I
from
mean, from the marine-
Sequaria. They collect
Yeah.
specifically for the pools.
Okay.
Yeah,
It
yeah.
used to be that Gore Green
Yeah.
and Phil Lambert would go diving.
Yeah.
Oh, they
Yeah.
do there?
Yeah.
Oh, I didn't know that.
But for things like in the collections or the labs, we try not to get stuff sort of going back and forth. Again.
Right.
But it maybe is an interesting point of conversation on that point of the collections over time and the information that the museums have because of that kind of data through specimens. That when you're at the tide pool or ocean station, bringing, I don't know, maybe you already do this, but bringing visitors awareness raised to the fact that the museum has many hundreds of thousands of specimens, and the reason is because exactly what Hugh was just talking about. Does anyone? I'm just thinking of that now, but
Sure.
so Bob,
Well,
You-
it's something I learned a long time ago, not just about this, but it was my question about longitudinal studies.
Yeah.
Because I have a university background,
Yeah.
and universities don't do that.
Yeah.
Don't do this
No, they do
Yeah.
short
stuff. And so it's a
term.
function that the museum has that the university does not. Yeah, I do.
yeah, great,
Not, not obviously. I don't walk up to somebody and say,
yeah,
"You know."
Yeah. But
But if
that's
it's
important. It's
if it's
important
kind
to
of
bring up.
contextual, yeah, I do.
And what I try and impress upon people is whenever I do tours is hey, look at these collections, they're really cool. The other thing that's important to know is that museums have, most people don't even know museums have these collections.
Exactly.
They think the museum is about what's on display. I would argue this is vastly more important than anything that's on display. Because
spoken like a true collection manager.
Because I'm a food collections or I'm a scientist. Yeah.
Do you
Yeah, what
ever
I-
do collection tours around here?
Yes, we
Like
do.
we do them. We work in the tower with Leslie Kennett.
Yeah, we do.
So we're putting the whole thing together from them to you.
Yeah, we do tours like that.
Yeah.
But what I really also try and focus on people is, or impress upon people, is that we do research here. I mean, people don't realize that as well. They think it's a university thing, but the kind of questions we can answer here and the kind of what the collections can do is, again, yes, it's as Bob pointed out, it's stuff that research that universities can't really do, or even government, or they rely upon our collections. There are some really exciting things going on in biology and biodiversity and all these kind of studies of climate change and how patterns are changing of animals and how animals may evolve differently, or there are these massive new huge databases of records of species and exactly where and when they were found. And I have a colleague who's done that and basically mapped every single brittle star specimen in the world, and then to show exactly where it was collected, when, and depth and everything, and looked at patterns of diversity of those and answered these really cool big questions. But every single one of those data points is a specimen in a jar on a shelf, and those big questions, those big environmental data sets that we're using to answer questions about invasives or climate change, they can't exist without these specimens on these shelves. Because otherwise, if I went out and collected and said, oh, I found a gumboot chitin and a crab and a so and so, and you have to take my word on it. Or I went out into the deep sea and I caught this, and then I just chucked them back. But you gotta, I identified them as this, and you can't really say otherwise because you can't see the specimens because I chucked them out. It doesn't work that way. You have to have these things, but on the shelves, vouchered for other people to see. That is the proof. That is the thing that underpins all of that data. And
Even Darwin knew that.
exactly, yeah.
Another question that keeps coming up, probably for everybody, is about the sea stars
Yes.
and the virus.
Yes.
So, do you have any new information?
Not really. I think there's some people who are narrowing in on something, and I think it's a virus. Yes, but they still haven't really worked out exactly what I mean.
There's a really good website called seastarwasting.org, which is University of California at Santa Cruz.
Yeah.
A lot of it is
Seastar
anecdotal
waiting?
because
Wasting.
Information.
just because, but it's probably the place that's, if you want to direct, I've directed a lot of people to it. I don't think any one of them has ever Googled it.
Yeah.
But nevertheless, I mean, it is
Yeah,
a reputable,
it's
reputable
still
website.
really striking,
Yeah.
and it's not Fukushima radiation that's causing it. It's not but it's most likely some sort of viral disease.
Do we have
populations are coming back. Populations are and there's also some evidence that this has actually happened before. There's some that happened in the 60s or this it's not the first time. It's probably the most dramatic. But it's not the first time this has happened either.
Is this nature's way of cleansing and keeping the species and things? Well, when you think about it.
I don't know if I wouldn't put it that way.
But you know what I mean?
I mean, nature is full of cycles,
Right.
like that, and it's. I wouldn't describe any kind of purpose to it. It's more that maybe there's enough of them around that maybe there's always this virus around, but if there's enough sea stars around that the virus can then quickly spread and actually knock them all out, and then they'll die and the virus population will fall and won't come back again for a long time.
Well,
So it's sort of cyclical. I always hesitate to describe any kind of purpose or any kind of like.
But we have to think a little bit.
Can we move
So my
back to where
question-
we
Yes, sure.
Yes.